So welcome everybody. We are going to have this session and this panel discussion where we are going to have opportunity to all the presenters, and I ask you already to put your questions on the Q&A part of this site. And so while people are still elaborating questions, I have some that I want to bring to you. And please, I'm gonna direct the question, but anyone who feel free to, who want to complement or something, feel free to jump in and collaborate. So I'm gonna start the discussion asking to Fred about the plug and play, right? And just before that start, I will ask everybody that is not speaking just to mute yourself, just to avoid any noisy. But Fred, let me just ask you regarding the plug and play that was developed by Simulation Plus, and a lot of people from the attendees were asking about how they can proceed with that. What is the requirement? At what stage of the development they should go and look for this kind of opportunity that is offered by Simulation Plus? Can you elaborate a little bit more about this?
Yeah, sure, Gustavo. It's a great question, and it's any stage you can use the plug and play compounds, right? Also, if you are in the early stage of the development of formulation, you can start to use the model to see if the direction that you use your formulation it's the right direction or not, right? So it's important you start to use earlier than later to play with those models, so then you can have a better direction to develop your formulation.
Okay, that's great. Thank you for that. Okay, let's go then to Xavier. So Xavier, I'm gonna put you a question that was made by the audience that we couldn't have time to discuss during our presentation, and it was from Rohit. And Rohit is just asked basically about the model you presented. Hi, Luiza. I just ask you to mute yourself, please. Xavier, the question was that the particle size distribution, the P particle size fitting, and eventually verified using batches of different particle sizes. Would this approach model hold for the solution affected by other formulation attributes like variation in hardness, variation excipient level? Would more variants need to be included into consideration then? You are on mute. You remember to mute and unmute also.
I remember to mute myself. I need to remember to unmute. So yes, it's a very good question. I think it depends if the mechanism of release is governed by, will be the same in different media. So when you run the verification, you typically predict from bottom-up approach the dissolution in other media, and so you have to make sure the inputs will translate into different in vitro dissolution. So for example, if there is a solubility limitation, this would typically if you have a disintegrating tablet, this would typically transfer to different media. Or if you have the impact of biosalt or the impact of surfactant, that can be captured. Now, looking at different variants, not only the size, if you have hardness of tablet, or if you have, overgranulation, compression, and any physical parameters to the tablet, this belongs to the batch. So, of course, the disintegration of that batch may be slower, and that may not be affected by the pH, for example. So this could still be, captured mechanistically by the PPSD model and transferred to other media. But I think at the end of the day, the verification step is very important to be able to apply this strategy. As for the Z factor as well, it's exactly the same thing for the Z factor. Any mechanistic model you have to verify before you can input and of course you can play on different variants. I had an example in the past. I worked on non-disintegrating tablets, so eroding tablets, which are far away from they are very big and they will just shrink from the surface. But the PPSD approach was very successful for the IVIVC for a project I cannot cite because it's secret, but it was working very well as well for non-disintegrating tablet. So that it's not only for disintegrating tablet, it can be applied, but you need to verify that it can translate to different in vitro conditions. And so for the non-disintegrating tablet, it was they changed the surface to volume ratio of the tablet to verify if the dissolution was comparable according to the surface area of the tablet, and that was the case. So it was then integrated in the model, and it was predictive of the. So again, it's a case-by-case study, but you can still use it for many different variants.
Yes, thank you very much, Xavier. So we're going to have a round of questions for all the panelists, and then in meanwhile, I encourage everyone to put your questions in the Q&A part, where I'm going to put to the presenters. So now I'm going to Jonathan and Maxime. So many of the questions, doubts, I think these have, is regarding how predictive this strategy and these models can be for the different drugs. We know that depending on the BCS classification, we have specific challenges regarding the predictability. And I'd like you to comment, it is the same approach for any drug, or is there any restriction, for example, for class 4 or anything like this?
Jonathan, you want to start?
I think you're not wanting to start, so I'm going to start it.
No.
I mean, there is no 100% guarantee. It's just to change the framework. I mean, if we look at most of the bioequivalent trials, people are looking at what they have done in the past and just replicate that. And the output of it is yes or no. Is it bioequivalent or not? And what we want to show is that there is another way, because if that's bioequivalent, how close it is, how many individuals did you, I mean, there is much more information in your clinical trial than what you, just this yes or no. And what we wanted to show is that there is a lot of questions on how to optimize your trial in terms of number of individuals, in terms of when to make the measurements, what is impacting, or should we look at a specific population, and that it is challenging from a decision point of view because it's not what has been done for the last 10, 20, or 30 years. So we want to put science to explain why it makes sense and why we should go for that, and that's a good example. This framework, I think, and I would like to give the end to Maxime, is kind of not specific to any drug, but has to be adapted to each specific issue you might have if you're on a specific drug or long-acting injectable, things like that, where the idea could be the same, but the way to solve it might be a little bit different.
Yeah, and just to add on that, I think the advantage of combining both approach is in the PPK model, you're being mechanistics. So whatever are the characteristics of the drugs, you may be able to integrate in the model. And then, for example, you've alluded to BCS class 4. So those drugs would be expected to have a very high inter-subject variability. So one of the key points would be to make sure the PPK model can reproduce that inter-subject variability before moving to monoliths. And it would be the same for the BCS based on the type of drug, compounds, dosage form, etc. As long as the PPK model is built in a mechanistic manner, the general framework should be the same. And then, as Jonathan mentioned, then maybe just tweaking here and there to adjust.
That's great. Thank you. Thank you, Jonathan, Maxime. So now Laurence. Laurence present a very succeeded case example of DDI and the possibilities on predicting DDI with modeling and simulation. And one of the questions that comes to mind is that we know that DDI is a topic that is under this harmony, right? We have Luiza here also, which is part of the discussion of the M12 group, which is pretty much now trying to incorporate these possibilities of in silico modeling to use in regulatory decisions, and also to support label inclusions and this kind of things. So you have a good case example, but I wonder if how acceptable has been this in your perception, Laurence, for the regulatory agencies, and how harmonized is this the way you have come to this result, and how it's going to be seen by an approval of a label of a product?
It depends on the different agencies, on the different authorities, because I said that we use the prediction for the different labels, so mainly the US and Europe. And for example, FDA did not accept the prediction for the labels, but they use only the results from the clinical study. Mm-hmm. And for and in Europe and in Australia, they approved the simulations, and for example, in the European label, we use the simulation for the recommendation. So it's really dependent from the authorities and the different territories, I think. And so it's not, I think we have to wait for more harmonization maybe. And maybe the new ICH guidance will address most of the issues. We can hope that.
We hope so. We hope we can get some of this harmonization, because it's science, right? And we don't expect any unnecessary barriers to things that might come with the same common understanding. Of course, each country has its own needs, but if you are talking about science and the capability of prediction, it's interesting to have this topic harmonized. And I think Luiza may comment a little bit about this. I don't know, because she's part of the group, and so there is some agreement on not disclosure. But my question, Luiza, to you is more related to the IQ consortium, right? So you presented some very interesting action that is moving agencies and industry, academia, and we know that it's really important to have all the stakeholders together, coming in a common understanding and a common ground to have decisions and to deal with the science. So I wonder, Luiza, what do you expect about this IQ, and what is the deliverables we are going to have for those who are developing a drug and wanting to go through this? How do you see, and how fast it's going to be?
Thank you for the question. We are seeing the IQ consortium, as was in the invitation, more as a scientific collaboration, and where we are becoming aware of the science behind BBBM, and we are discussing this in a very unique moment where the topics are being raised and the science are being developed. And this is very important for our agents like Visa, that is not always in the same page of the science, in the same, we don't have enough experts as the other agents like FDA. So sometimes we get on the harmonization where the science is more mature, like for PBPK for DDIs in N12. So PBBM has been different as we are being able to go with the discussions. And of course, there is discussion about other agents like 1 decade ago, but we are being able to discuss this. From a regulatory perspective, we're going to have to frame this according to our reality and our specific demands, and maybe the reliability that I mentioned would be very valuable for us to see if we ask the right regulatory questions and we accept PBBM as a tool to answer the right regulatory questions that have the patient as the focus, as the interest. So nowadays, we don't see a barrier for analyzing PBBM. We are gaining capacity. This collaboration with academics and also with the discussion with software development and among regulatory agents have enables us to do diligent assessments. We are gaining confidence with this, but also we see this. We should now put this in a bigger picture. As I presented, we had only 6 cases in 2 years. So from a risk perspective and from the agent's perspective, it must not be in the same priority as other issues. But what I can say is that there is no barrier to discuss a specific applications. But definitely, we must involve all the other stakeholders in the agents, other stakeholders in our local industry, and connect these initiatives with other modeling simulation discussions.
That's great, Luiza. I don't know if Fred wants to also add. I know Fred is also involved somehow on this, right, Fred? And Xavier also, please. Oh, okay. So we have some questions here regarding the. So Raja made a question that Jonathan already answered regarding most of the models are focused on oral absorption, and what about the other possible pharmaceutical forms like long-acting injectables? Jonathan answered, but I don't know, Maxime, maybe you want to comment also on this, and Jonathan also to comment, please.
Yeah, sure. So I think what is important to keep in mind is when we talk about PBM, PPK models, the key word behind it is really mechanistic models. Whatever the route of administrations, as long as we describe the mechanisms involved on top of the dissolution, but what could happen. So for example, for LAI, the local inflammation, the precipitation, etc., that occurring at the depot site, we will be able to build PBM, PPK model for those route of administrations. The difference is that right now, for locally acting and other products, the model are more recent compared to oral absorption that is fairly well understood. What is interesting in the case of LAI, but all of the other route of administrations, we currently have collaboration with the FDA to improve the PPK model for example, ocular, dermal, LAIs, and exactly to perform what the question alludes to, like how to build the PBM PPK, so we can make the link between in vitro characterizations and in vivo behavior formulation, and then why not extend to simulating population, doing virtual BE, and we could plug that as well and go back to the comment Jonathan has made, is combining those with population PK approach to design clinical trial for those specific drugs, etc., etc.
Yes. Jonathan, do you want to add something or?
No, just to complement Maxime's answer. Again, I mean, if we are able to start the modeling, we can try to have, as we did for the example, using PBPK or PBBM as a target to say, okay, that's what we think is the reality, and to use the population modeling to say how we transform that to a real clinical trial, which is especially challenging for the long-acting injectable, because if you are looking at crossover trial, if you want to make sure that between the test and the reference, you have sufficient time, it can take ages. So we have to understand exactly what is needed from a statistical point of view to make sure that the information we gave to the regulatory agencies are safe and understandable by all the stakeholders. And that's what we've done in a specific collaboration and a project with the FDA on that specific topic.
Great, thank you. So Marcelo asked something regarding the acceptance of regulatory, and I want to leave this question. Xavier answered some aspects, but I want to take this opportunity also to ask more broadly. This question of Marcelo was considering that the model can predict performance in vivo with formulation changes. The question is how regulatory agencies and how experienced, this is something that came also from Fred's session, how regulatory agencies, how many cases of success we have with regulatory agencies. Maxime mentioned some collaborations with FDA for some cases. We have Luiza also here from Brazil. European, Xavier has experience with European. So I think one of the most one of the most common doubts is, will I be able to use this to submit for formulation changes, for justification of specification? And this is something that was presented, but I just want for you to say again, to reinforce this, how acceptable, what is the percentage of success we are going to have if we use these tools for regulatory submission? So Fred, please, if you can start.
My opinion is the rate of success. It will depend the data, right? The output of your modeling, of course, the D plus and G plus can guide you. But I mean, you have a correlation with your formulation and the clinical data or pilot. So if you have this, then your chance to be accepted with the population will be much bigger than if you don't have.
Yes. Xavier, please.
Yeah, I agree with Frederico. It's all data driven, so you need to you need definitely to have a strong model validation. And what is important in your model validation is to use the same scenario that you want to use for the end model use. So, for example, in the question from Marcelo, he was mentioning DDD plus to predict in vitro dissolution of following formulation changes, and that is perfectly doable, but it's another step that you add in terms of prediction. You know, there's the PBBM, and typically we use it with measured dissolution. Now, if you want to predict dissolution on top of predicting PK, that's something you can do, but you need to demonstrate to authorities that the model can predict all your clinical experience. So you need to show on meaningful variants that the model can predict dissolution and can predict PK as well. And it's really how you build your validation strategy. It has to be exactly the same approach you will use eventually to interrogate the model itself. So you can do that definitely. What I've seen from my personal experience, when you introduce a new technology, you need to spend some time with regulators to explain how this new technology works, what are the limitations, what are the opportunities, show some examples, show where it works, where it doesn't work, give some limitations, because that will actually engage them with scientific discussion and really show that you have a sound approach to the new technology, to the approach that you're proposing. And I've seen it with the PPSD. When I started this back probably 3-4 years ago, there was lots of discussion with FDA initially, and to be able to explain what it was, to explain how we verified it before introducing it. And it's a new concept, so you have to reassure people, say, introduce it, explain it. And I think that's the same for any new technology, any new modeling approach. You need to spend the time and explain the how you've done things and how you validated things. So I would say there's no magic number. It's it depends on, as Laurence said, it depends on the agency. Sometimes you have no discussion. You even wonder if people have read the model or looked at it. But sometimes you have many discussion, and that is really interesting because you realize that people have read the model, have tried to understand it. They come back with interesting questions. So it's also making you progress on how to present things and how to do the actual dossier. So my message to people is to say, don't be afraid with new technologies. Just go. Submit, discuss, do TCs, answer to information requests, and the more you will discuss, the more you will explain. I think you will make your science progress. You will make also the use of modeling and simulation progress, and that's for the benefit of patients eventually, because these models bring mechanistic understanding, contrary to a clinical trial which may be successful by luck or unsuccessful by luck. Here you can run tens of virtual trials and really understand what is going to be the limiting factor.
Yeah. So Luiza, I think we have time for your comment, and then we have to close our session.
Okay. Yes, I think I agree with all that Xavier and Fred have said, and also should emphasize that from the agent's perspective, it's very important to have assessment of the model, considering the consequence and the influence of the model, to have assessments that goes beyond the scientific details and put the risk on perspective. It's very important. And this collaboration is very important for we to have this scientific discussion, to leverage our knowledge on this, and to have the regulatory perspective is very important, because we would be cautious with applications that intend using of a new technology that has its uncertainties, like model for normal scenarios, like for registering a generic drug product based on VBE will not be an acceptable application at this point of time, and maybe in the future. But now we tend to follow what more mature had discussed and decided. So the reliance and the being connected with other agents and other scientists is very important for the regulatory decision that should put the patient in the focus.
Well, I think that also answers Rebecca's question. And with that, time is up. Thank you very much for a very interesting discussion. Thank you for the opportunity for us to learn. Thank you for the audience to be here, to pose the questions and to discuss. And have a great day. And thank you very much.